So hello everybody, my name's Jeff Coxon and I'm from Skylab, and what we thought we'd do today was tell you how these work, medicines. And how do medicines work? Well, they contain something called a drug molecule, API, active pharmaceutical ingredient. And in this case, it's paracetamol. So we thought we'd tell you how these work and also how you invent a new drug molecule. It's very, very complicated and it takes a long time and a lot of money. So we thought we'd go through it pretty quickly just so you can get an idea about how this works. Now this won't be completely detailed, it won't be completely exhaustive, and I'm sure there'll be different ways in which you can explain it, but I'll try and do it as simple as possible. There will also be a lot of big, long, fancy words as well. So bear with me, I'll try and explain them to you. So, right, so this, now paracetamol, it's a very, very common drug that you'll find in the house. It is bought in a chemist or in supermarkets. It's called an OTC drug, an over-the-counter drug. Okay, that means you don't need a prescription to get it. So, in order to think about or start this explanation and the story about how a drug works, we need to think about what you do with a medicine. So let's look at what you do. So when you open the box, you take out your blister pack of your tablet or your capsule, and you pop open the blister cap, your blister open, and you have your capsule or your tablet. In this case, it's a paracetamol tablet. Okay, and then what happens next? Well, normally the majority of drugs are what we call oral drugs. So they're tablets, you put them in your mouth, so that we call them orally available, or an oral mode of drug delivery. So you're going to take this via the mouth. So this is where we need to think a little bit about the journey of a drug, to think about what it actually does. I'm going to share this screen with you. So let's have a look. So here we are, right. So this is us, human body. I'm going to make this a little bigger so we can see this. I'll move it in a little bit here. Right, let's think a little bit about this. Right, so here's our body and here's our mouth. Remember, we have our tablet and we're going to swallow it. So this is what happens with your drugs. It starts this journey. So it goes into the body and it goes in the mouth, down through back of the throat, the esophagus, down the windpipe, down to esophagus, and into your stomach. Okay, so it's right in here into your stomach. So the drug molecule goes in, and when we think about the journey of a drug, we need to think of it in simplistic terms. So simply, it goes in, which we've done with the stomach, it goes about the body, and it goes out. So it goes in, about, and out. And it leaves your stomach either in your urine, in your pee, or in your feces, in your poo. So that's how it gets out of your body. But getting out is via your pee or your poo. Going in is normally via your mouth. But about is where it gets quite complicated. So it goes in and about and out. And when we think about drug molecules and how they work, we think about 4 letters, A, D, M, and E, or ADME. That stands for absorption, distribution, metabolism, and excretion. So in, about, and out. And this is how a drug molecule works. So we take the tablet, the paracetamol, or it could be any other kind of drug molecule, take it orally, it goes into the stomach, and it absorbs. So it absorbs either in the stomach or in your intestine. So the drug molecule can absorb from the intestine, from the stomach, and it absorbs into your blood. And it absorbs into your bloodstream via something called the portal vein. And once it absorbs from the stomach or from the intestine, absorbs and goes through the portal vein. And the first place it passes is your liver. And the liver is here. And when the molecule goes through the liver, it comes out of the liver, and it's what we call, or what has happened is something called first pass metabolism. And from the liver, it then goes into your systemic circulation and goes around your body. It distributes around the body. So first thing, D, the distribution, it distributes around the body after it goes through the liver. And the liver metabolizes the drug. That's where the metabolism comes, the M, the metabolism. And then when it distributes and goes past the liver again and again, it gets metabolized or broken down because the body wants to break down the body or break down the drug molecule, and does the same thing, exactly the same thing with your foods, with your curries, your burgers, your beans and sausages, whatever you have. It breaks after absorption, it gets broken down, and your body breaks those parts down as well. And your liver metabolizes them and breaks them down. And your liver breaks them down into molecules that are smaller or more water soluble. So they will help to be excreted in the urine, or it will make them bigger and it will pass them through your bowel, through your feces, and you will poo them out. So the job of the liver is to convert the molecule, break it down so you either pee them out or you can poo them out. And when you pee your molecules out, you pee them out via your bladder and your urine. If you want to poo them out, then when they get converted so you poo them out, they go via your bowel and your anus and your lower intestine, and you poo them out. So that's how the body goes, or how the body breaks down drug molecules via the liver and then the kidney. So it's in, about, and out. Absorption, distribution, metabolism, excretion. And whilst the body is doing all of that to it, and it's distributed and being metabolized and excreted, the body is going around the systemic circulation. And as the drug goes around to systemic circulation, it goes into every part of the body. It goes through your muscle tissue, your lungs, your arms, your brain. And depending upon how the molecule or the drug molecule looks like, in other words, the molecular structure of the drug molecule, it will go to different parts of the body better than it will go to others because of the way the chemistry makes up the drug molecule. Because a drug molecule has a particular shape. And that particular shape of the molecule, the molecular arrangement, its molecular structure, governs how a drug molecule works and where it goes to. And then we think of two letters, and they are P and D, and that stands for pharmacodynamics, what the drug molecule will do to your body. And that is when a drug molecule will act at a particular drug target. For example, it might work in the lungs against things called beta adrenoreceptors, beta 1 or beta 2 adrenoreceptors in the lung. Particularly the beta 2 adrenoreceptors. And so, for example, if you had asthma attack, you'd have salbutamol in an inhaler. You inhale it into your lungs. It would then act and work on a particular adrenoreceptor. That would help dilate your bronchi and you'd breathe easy. So that's an example of pharmacodynamics. Or it might work against a bacterial cell and break down, like penicillin will work against a bacteria. It would destroy the cell wall of the bacteria and would kill the infection. Or we might have a drug too that might target particular enzymes that will reduce blood pressure, for example. Or antiviral drugs, which might help destroy the virus. So when they work in that way, they have what we call a particular mechanism of action, and that's what we call the pharmacodynamics. When the actual drug molecule itself, the API, the active pharmaceutical ingredient, it gets around the body and it finds its way a little bit like a lock and a key. So for example, the key will fit a particular lock. It will interact with a particular part of the body, particular what we call an enzyme or a receptor, and that will exert a particular pharmacological effect, which is lowering blood pressure, stopping an asthma attack, killing a bacteria, killing a cancer cell, etc., etc. We call that PD, pharmacodynamics. And when we were talking about the body breaking down the drug molecule and it being broken down so it leaves the body, we call that PK, pharmacokinetics. So it's a balance between the drug molecule working against the target, which we call pharmacodynamics, and the pharmacokinetics of getting the drug molecule out of your body and distributing around the body. So it's very, very complicated. So it's a real balance between getting the molecule to work where it wants to and the body to get it out. So at the right particular dose and at the right particular dosing interval. So for example, we're talking about our paracetamol, which we take every 4 to 6 hours. That's the normal dosing interval for you taking a paracetamol drug molecule. So your medicine paracetamol every 4 to 6 hours, no more than taken 3 times over a 24-hour period. That's because we want to make sure that the body stays in the system long enough but doesn't cause any nasty toxic side effects. So we have to make sure that we take the right drug in the right doses, in the right amount at the right time. And that's very much about the pharmacokinetics and studying that drug molecule, getting the dosing right. So you got to balance all of that right to be able to have your medicine to work properly. And this is the reason why inventing a medicine is so extremely difficult and complicated, because we have to balance everything in the body against how to make a molecule, invent a molecule, and make sure it works in a right place and not be toxic and not do something that it shouldn't do, because you want drug molecules to be safe. It's very, very complicated. Excuse me. So now we know what happens to a drug and how a drug molecule works. We think about how we actually invent it. I'm going to go back to me. So I'll just stop this screen share. Bear with me. So we'll go back and we will stop the screen share. Let's go back to Zoom. And we're going to go here and we're going to stop this screen share. Bear with me one moment whilst I stop this. So stop the share and back to me. So this little tablet is extremely complicated, and we now know about its journey. So how do we actually invent one? So it often starts at a particular point, either because we know something about medicine, we know how a particular molecule or an existing drug might work, or when we think about, say, for example, how we invented asthma drugs, so drugs that we used to treat for asthma attacks, there is some form of starting point. And we think about asthma, the actual invention of asthma drugs, like, for example, salbutamol, all goes back to the 40s and 50s when we were starting to understand about science and look about what certain molecules or what certain extracts of plants or animals would actually do. And for something like salbutamol, which is a starting point of, which is an asthma drug, it all went back to a drug molecule called adrenaline or epinephrine. And it went back to observations that biologists and pharmacologists made when they decided to mash up the adrenal glands of sheep, and they found a molecule which we now call adrenaline from the adrenal glands. And once chemists were able to work out the structure of that molecule, they worked out that some effects were good and some effects were bad for the body. So they made more and more and more molecules, different versions of the drug, tested them, saw they did, make different versions, tested them again, and so on. And there's an iterative process between making and inventing drug molecules, testing them, seeing what they're doing. And that is the start of the drug development process, what we call the discovery phase of inventing a medicine. Now, nowadays, the last 20 or so years, that has been heavily dependent upon a lot of technology, a lot of modern techniques, and a lot of genetics. And that is the early stage of drug discovery. And once you've found a molecule and you've tested it against a particular cell you're after, maybe a cancer cell, for example, or you're testing it against different kinds of fungi or bacteria or viruses, you test them in the laboratory, you make lots of different versions, And then you want to make sure that they are safe. So you test their toxicity in a laboratory. We call that in vitro testing. So that's within the test tube or within glass. So we test them very much in a lab environment. And we test them against a whole different range of cells and enzymes, those from the liver, those from other cells, to make sure that we at least know that they're not going to attack cells that are in us, in humans. So we at least have an idea about that, and we get an idea about how they may or may not work. We test them against to make sure that they're potentially not cancerous, or that they may not disrupt the rhythm of the heart. And once we've done that, we then have to put them into animals. So normally into rodents like a mouse or a rat. And then if that's safe, we then go into guinea pigs or rabbits, and then possibly into higher order animals like a rhesus monkey or a macaque monkey. And we have all of that data about how we work in animals, how they work in cells, and we understand how they work. So we understand about their mode of action or the site of action, and we have a lot of data. And we take that data, and that data is taken to, in the UK, it's the MHRA, or Europe, it's the EMA, or in America, it's the FDA. And these are the governing licensing or regulatory bodies that will give a license for the use of a drug in humans. And eventually, you have to go through a phase of human testing, phase 1, phase 2, phase 3 testing. When after phase 3 testing, a license is given, and then it can then go into the clinic. And normally phase 1 testing is when that drug molecule that you've had all that testing from in the lab, that you have a small group of patients, normally healthy patients, and you test its pharmacokinetics, its pharmacodynamics, and you test its dosing range. When that's happy, or you're happy with the data from that, let's say if you go back to the MHRA and you go to what are called phase 2. Phase 2 are normally into larger populations, maybe 200, 300 patients. This time with the disease indication, you want to have an idea of the efficacy of the drug. So is it going to work against the disease in which you've designed it for? So for example, new antiviral, or new anti-asthma drug, or new weight loss drug, or new antibacterial, new anti-TB or anti-AIDS HIV drug, for example. So you test it in a larger population within particular, which actually have that disease. When you're happy with that, the data is good, then the MHRA, for example, would say, right, you can now go into a much larger population. And that's called a phase 3 trial in different sites and different locations. And when the data is satisfied, or the data is satisfactory, then a license is given, and then it goes into the clinic. And then normally a phase 4 trial may occur, or as normally a sort of a post-study trial. And when the drug is in use, and it goes into thousands upon thousands or millions of patients, monitoring of the drug is studied for potential side effects or adverse drug interactions over when it's actually in use. But constant monitoring of the drug is always done. So that happens, and that process from the discovery phase all the way to when you get into phase 3 trial in the clinic can take 10 years and costs hundreds of millions of pounds. So we have the discovery phase, we're very much lab-based, very much into the clinical base. So I am very much, my perspective is from the discovery phase, when we learn how to invent molecules, and that early part of the discovery process. And we mentioned about salbutamol as an example, how to invent it, make the drug molecule itself, and some of the more the sort of technologically advanced techniques that we use today. So I'm going to give you an example of that, and I'm going to go and I'm going to share the screen here again. You bear with me. So I'm going to share the screen. Here we go. So I'm going to share this. I'm going to show you what the sort of technology and the sort of software that we use to illustrate how we might invent a drug or see how a drug works, so we can learn, so we can work out what we need to do to a molecule to invent more of those molecules, do the chemistry, test them, see what happens. And this is salbutamol. So this is the molecule that I was talking about to treat asthma. And if we look here, this is some software that we use to look and be able to visualize drug molecules. And if I just turn that off, So this is this. Oops, bear with me. I'll turn this back off again. So this is salbutamol. This is the drug molecule here. That's what the drug molecule looks like. And it has various different components to it. So that's a drug molecule. So that's an API. That's an active pharmaceutical ingredient. So that's, if you like, the key within our lock. So I'm going to put the lock back on. Now, the lock is something called an enzyme. And an enzyme is made up of amino acids, hundreds and hundreds of amino acids in a particular sequence. That sequence is determined by genes. And the enzyme, in this case, our target, is the beta adrenoreceptor. And what salbutamol does is it binds within what we call an active site of the enzyme. And the active site is, for example, or we would say it's the key going into lock. And the protein made up of amino acids and the drug molecule of salbutamol, if their particular chemical modification, or their chemical structure is very much compatible, then the drug molecule binds and sits within the active site. And when it does that, it stops a series of what we call pharmacological or biochemical interactions, and that will stop your lungs from, or your bronchi from being very, very constricted, and it'll dilate those bronchi, your bronchi, and you will breathe easier. So it sets up a whole series of different biochemical transformations and leading to a particular therapeutic effect, all because this molecule, in our case salbutamol, binds within the pocket of the beta 1 or beta 2 in this case adrenoreceptor. It's called, in the case of this, it's called, if salbutamol, it's called signal transduction. So it stops a particular signal transduction events happening so that your bronchi dilates and you can breathe. Okay, very, very complicated, but that's essentially what it looks like. And the way that this molecule binds pharmacodynamically is if we look at, for example, all of the bonds. So put all the bonds on here, some hydrogen bonds, for example. And these hydrogen bonds, when this works, here we go. So all these interactions are what make your proteins and your enzymes bind together. And these interactions are all because of the electronic configuration on the drug molecule and the proteins here. So it's very, very complicated. That's how we invent, or we'll be able to look and see how molecules work. So that's how the drug molecule acts at the target. That we call the pharmacodynamics. And similar things will happen pharmacokinetically with the enzymes that metabolize drugs as well. So it's a very, very complicated way in which we're able to do that. So that is a whistle-stop tour of how drug molecules work, how you invent a drug molecule, and I use salbutamol as an example and paracetamol. So next time you have a drug, you take a medicine, think about what goes into that tiny, tiny little white tablet. Very, very complicated. So that's the whistle-stop tour. So I hope you enjoyed that. I hope that gave you a little bit of curiosity to go and think more about drugs and medicines, and pop back to either our Twitter page on Skylab or the Skylab Facebook page or the Skylab website and see what we're doing with STEM, and stay safe, and we will see you soon. Bye-bye.